Hello students, welcome to the EPG Pathshala. I am Dr. M.N. Gupta, Emeritus Professor from Department of Biochemical Engineering and Biotechnology at Indian Institute of Technology, Delhi. Today we are going to talk on the module called Cytotoxic cells and cellular immunity from the paper on immunology. Some bacteria enter animal cells. Viruses also infect cells. In such cases, cellular immunity is required for protection. In some fungal diseases as well, cellular immunity plays an important role. We have already learned that T cells are the ones which mediate cellular immunity. Let us look at that in detail and see how these cells work in concert with the rest of the immune system. So, the objectives of this module are to understand the role of cytotoxicity and cellular immunity in various diseases, also to learn about how both innate immunity and adaptive immunity deal with intracellular pathogens. Furthermore, also to learn about natural killer cells and cytokines, the role of the cytokines in more detail, and to learn about thymic education. So, the concept map essentially looks like this, that we are going to focus on intracellular pathogens and then see that how thymus educates the cells, and some of them T cells, and some of the T cells become cytotoxic T cells, that is the TC cells, and along with innate immunity part, which here is represented by natural killer cells, they create a synergy to deal with intracellular pathogens. These TC cells secrete cytokines, which in turn also influence the behavior of the TC cells. Similarly, these cytokines secreted by TC cells affect innate immunity cells called NK cells, and NK cells themselves also secrete cytokines. So, there is a lot of cooperativity in this process. The discovery of cellular immunity was made when it was realized that some properties could not be transferred between animals through serum. A delayed hypersensitivity response to tuberculous bacillus protein preparation could be transferred to a normal animal by cells from the spleen, lymph node, and a sensitized guinea pig. At one time, it was believed that thymus in mammals discharged the functions of both bursa and thymus in chickens. This should help in understanding the excitement which resulted from the work of Jacques Miller who subjected mice to neonatal thymectomy. Warner and Salzberg showed the respective functions of thymus and bursa in chickens. Graham Mitchell and Jacques Miller soon thereafter showed the role of T cells in antibody production in mouse. Humoral response is able to deal with extracellular forms of pathogens and the toxic products. Even these we have already referred to the importance of the Th cells, which are part of the cell mediated immunity. Some pathogenic bacteria and parasites and all viruses invade host cells. These intracellular invaders are not accessible to antibodies, hence the need for T lymphocyte which mediate cellular immunity. One kind of T cells called CD4 T cells defend against bacterial infections which are intracellular in nature. Mycobacterium tuberculosis and M. leprae grow inside the membrane-bound vesicles of macrophages. These organisms are responsible for causing tuberculosis and leprosy respectively. The vesicles containing these bacteria do not fuse with the lysosome of the macrophages. Th1 cells make this fusion possible and activate macrophages to cause phagocytosis. A Th1 cell recognizes the infected macrophage, releases cytokines which facilitate the fusion and bactericidal activity of the macrophage. In absence of Th1 activity, these bacteria go on multiplying inside the vesicles. The other examples of intracellular pathogenic bacteria are Salmonella typhi and the Brucella species, which also have vesicular residence. There are other bacteria such as Chlamydia, Rickettsia and Listeria species which replicate in the cytoplasm of host cells. So, one way of classifying intracellular pathogens is according to their cytoplasmic or vesicular nature. It should be made clear that virus entry into cells at initial stages of infection can be prevented by neutralizing antibodies released by B cells. The production of these antibodies is facilitated, however, by Th cells. These neutralizing antibodies are directed against viral protein coat, which is highly antigenic. However, once the virus entry in the host cells take place, these neutralizing antibody cannot reach it. The role of T cells in protecting against these pathogens deserves further attention beyond the simple outline which indicates the importance of T cells. We will now describe immune mechanism and indicate the importance of cell mediated immunity in such cases. Some bacteria which do not invade cells exert their pathogenicity via epithelial attachment. These generally release toxins inside the cells. Corynebacterium diphtheria, Clostridium tetani, Vibrio cholera, GPA staphylococci, which cause sore throat, are such pathogens. The second class is those of intracellular bacteria, which already have been identified. Many bacteria in fact show mixed behavior. Staphylococcus aureus and Clostridium perfringens exemplify this behavior. There is some limited local invasion. In such cases also, cell-mediated responses are required. The bacteria which have capability of invading the cells also have to clear non-specific barriers which have been discussed earlier and may be recalled again in this context. Commensals, that is organisms occupying a specific niche in various parts of the body, play an important role here. Vagina epithelium secretes glycogen which commensal bacteria metabolize to lactic acid. Consequently, the acidic pH discourages many infections. Infections by Candida and Clostridium difficile occur more easily when commensals are destroyed or disturbed by antibiotic treatment. Both prebiotic and probiotics can help in re-establishing the protective flora. Many components of innate immunity do play a protective role. After intracellular infection by the bacteria, these act independent of B cell and T cell mediated immune mechanisms. Many organisms, for example the non-pathogenic cocci, are eliminated via these mechanisms without adaptive immune mechanisms coming into play. Complement activation via alternative pathway independently or by facilitating phagocyte infections can destroy the infected cells. Macrophages release cytokines TNF and IL-1 which not only activate phagocytic cells but also promote their entry into tissue at the site of infection. Data with murine cells show NK cells in response to microbial components and TNF in turn release IFN gamma which further activates macrophages. Mice Listeria monocytogenes infection, even if these have severe combined immune deficiency which prevents T cell maturation. Depending upon a particular infection, its pathogenicity and severity, T cell mediated responses often become necessary. The cytokines released determine how T cells direct various effector mechanisms. Thus, decision is made by subtype of T cells called Th cells, which stands for T helper cells. The three key effector mechanisms involve Tc, cytotoxic T cells, antibody plus mast cells and eosinophils, macrophage activation and delayed hypersensitivity. Thus, decision by Th cells is very critical. The third option is protective in leishmania, whereas second option is hurtful. In the murine system as well as in humans, different forms of T cells release different cytokines. So the sequence of decision making involves cytokines at two stages. This is shown in this slide. Cell mediated cytotoxicity operates through not only Tc cells but other lymphoid and myeloid cells as well. It may bring some clarity if we rather look at the way these cytotoxic cells bind to the target cells. Specific antigen derived peptides along with MHC proteins are targeted by Tc cells via their TCR. Receptors on NK cells recognize some antigenic determinants like on tumor cells. AB-Ag complex, it could be viral antigen or infected cell which is a part of this complex. These complexes are recognized by Fc recognizing receptor on K cells. This is called antibody dependent cell mediated cytotoxicity which was explained earlier. Let us look at the interaction between Tc cells and target cells. Tc cells have a very complex interaction with the target cells. It involves multiple linkages which firm up the binding between the two cells. This action is called antibody independent cell mediated cytotoxicity. To distinguish it from ADCC, it should be however very clear that Tc needs to see the antigen on the surface of infected cells. That's a critical design which enables a Tc cell not to start attacking uninfected cells under normal circumstances. Thus, an important role of Tc is in killing virus infected cells so that the host cells of the virus are destroyed and virus multiplication stops and viral infection spread is halted. Tc cells action is also different from Th cells, which cooperate with the humoral response of B cells. That is another reason why TC cell mechanism is called antibody independent cell mediated cytotoxicity. Many times during our discussion, we have used words like cytokines, lymphokines, and at times mentioned specific interleukins. This may be a good time to bring clarity about these terms, as we will continue using these in even in later modules and quite frequently. The term lymphokines refer to the mediators released by lymphocytes. Interleukins, along with the number assigned to each, for example IL-1 or IL-2, was an attempt to create a system of nomenclature for mediators with an implication that these were part of interleukocytes, that is white blood cells. With further work, it has been found that interleukins functions cover even a wide range. Cytokine is a more general term. It includes lymphokines and interleukins. Cytokines are peptides, often glycosylated. Excessive release of cytokines can be harmful and can result in severe tissue damage, shock, and circulatory collapse. In the pre-DNA era, in areas like immunology and molecular biology itself, several mediators, effectors, and factors were reported. Isolation and characterization of these molecules was difficult, as these occurred in small amounts and enzymology did not have tools to separate these factors at micro level. In many cases, later on it was found that different labs were actually often looking at the same molecule. In some cases, these studies were being made in different contexts. Thus, cachectin cytokine reported in the context of weight loss in chronic infections turned out to be same as TNF alpha. IL-6 was separately identified as hepatocyte stimulating factor, monocyte inducer, beta-2 interferon, a B-cell stimulatory factor, and by few other similar descriptions. Production of cytokines by cloning helped molecular characterization and brought some clarity. However, cytokine functioning in vitro should not be straightaway extrapolated to in vivo physiological effects. This is because cytokines can act in synergy, in sequence, and even antagonistically. IL-2 and IFN gamma together aid cell activation. IL-1 and IL-2 induce different receptors which may be required in a cascade reaction. If IFN gamma and IFN alpha have opposite effects on production of IgE by B lymphocytes. The activation of human macrophages by IFN gamma is opposed by several cytokines like IL-4 and TGF-beta isoforms. In fact, cytokine effects can be inhibited in vivo by three different mechanisms. The first mechanism is similar to the phenomenon of competitive enzyme inhibition. Cytokine analogues compete with the cytokines for the receptor. Thus, the IL-1 inhibitor competes with the IL-1 for the receptor. Extracellular domains of cytokine receptors come apart and become free, as these were the components of the receptors with which cytokines were to bind. Free complexes of cytokine receptor fragments are formed, which tie up cytokines in interacting with the target cells. As mentioned and exemplified earlier, two cytokines can oppose each other function. These will have different receptors. These three types of cytokine inhibitions are pictorially depicted here. This slide shows how complex is the physiological role of the cytokine and how cytokine actually forms a network. While the few effects of IL-6 were known, work with transgenic animals in which IL-6 was overexpressed showed up its role in production of platelets and megakaryocytes. Cytokines not only are part of cell-cell communications, these are important for communication between different organs. A parallel can be drawn between nervous system and immune system with their common feature of learning and memory. Both have different communication systems but with some common features. Both systems in fact also interact. Now it is known that the several cytokines like interleukin 1, interleukin 6 and TNF have effects on the hypothalamus and pituitary organs as well. Let us now continue our discussion of cells other than TC cells which mediate cytotoxicity. Having learnt about what cytokines are, our discussion will continue to include even in more detail the role which cytokines play in cytotoxicity. The NK cells to which we have referred earlier are also called null cells and third population cells. These constitute about 5 to 15% of blood mononuclear cells. These do not have receptors like BCR or TCR, but recognize T cells as well as mononuclear phagocytes. Many of these morphologically appear as large granular lymphocytes, sometimes simply referred to as LGLs. NK cells do have many receptors which include CD16, which is a receptor for Fc fragment. They also have receptors like CD2, CD69 and lectin-like receptors which allow these cells to bind to wide-ranging cells. Killer immunoglobulin-like receptors called KIR are a family of receptors which are also present on NK cells. These recognize MHC molecules. Killer immunoglobulin-like receptors occur in two forms, one with the ITIH motif and another with the ITAM adapter like motifs in their cytoplasmic tails. These motifs have been explained in the signal transduction chapter. The number of KIRs varies from individual to individuals. Not all NK cells express all the receptors. Each NK cell has a set of receptors which enable it to monitor change in one group of MHC molecules. Thus, NK cells population is able to monitor change in the pattern of MHC expression by the cells of the host animal. The surface receptors on natural killer cells are listed here and their function is also shown in the right hand side column. Many viruses try to avoid immune system by downregulating the expression of MHC molecules of the infected cells. How and why this may work will be clearer only when we discuss MHC molecules in a later module. NK cells detect this via KIRs and kill such infected cells. This is so important that even some T cells may also express some KIRs after activation. The NK cells activity reflects the balance of signals from ITIM and ITAM, which in turn are responding to change in the pattern of MHC of host animal cells. Lectin-like receptors are a family. Each member has two polypeptides, NKG2 and CD94. Again, these receptors are present on all NK cells and some TC cells. These receptors recognize leader peptides of MHC molecules. This also serves the purpose of detecting loss of MHC molecule by a cell. These receptors also are regulated by their cytoplasmic tails. Some toll-like receptors on NK cells are also present which aid cytokine production. Two mechanisms help NK cells in not killing normal uninfected cells of the animal. This regulation operates via both activation and inhibition mechanism, as explained while discussing the role of the KIR receptors tracking change in the expression of MHC molecules on animal surface. Lectin-like receptors are calcium-binding C-type lectins which can monitor carbohydrate ligands on the animal cells. Intracellular bacterial or viral infections result in changes in expression of the cell surface glycoproteins. NK cells provide early defense to intracellular pathogens like herpes virus, protozoan parasite, and bacterial pathogen Listeria monocytogenes. NK cells, even if untriggered, can kill appropriate cellular targets in vitro. However, cytokines IFN alpha, IFN beta, and interleukin 2 produced in early phases of infections increase the NK cytotoxic activity by 20 to 100 times. IL-2 plus IFN alpha induce NK cells to secrete IFN gamma, which is critical in immune defense before TC cells secrete IFN gamma. Mice lacking B and T cells resist Listeria monocytogen infection initially. Later, NK cells and IFN alpha, IFN gamma levels lowers and mice due to absence of adaptive immune responses die. We have referred to interferons several times. Viral infections produce interferons IFN alpha and IFN beta. Interferons are glycoproteins and double-stranded RNA induce their synthesis both in vivo and in vitro. This RNA is not present in the mammalian cells but is either part of the viral genome or produced during their cycle of infection. IFN alpha and IFN beta bind to a common interferon receptor on both infected and neighboring cells. This receptor uses genus family tyrosine kinase mediated signal transduction pathway. Signal transducing activator of transcription, STATs, induce synthesis of host proteins which inhibit viral replication. Both NK cells and TC cells have similar mechanism of cytotoxicity. In both cells, there are lytic granules. These modified lysosomes have perforins and granozymes, which are 3-serine proteases. These trigger a cascade, an activated caspase CPP32, which in turn activates a DNase. Degradation of DNA leads to apoptosis. The size of the thymus increases till puberty and thymus gradually atrophies thereafter. Some thymic function continues even in old age. The number of T cells which mature out of thymus gradually decreases with age. With age, there is an increase in the IL-6 and MCSF, that is the macrophage colony stimulating factor. These all decrease in IL-2, IL-10 and IL-13, and there is no change in IL-7 and IL-15. Thymus interacts with the endocrine system. Removal of the thymus results in decrease in levels of pituitary hormones and atrophy of the gonads. Conversely, removal of the pituitary leads to atrophy of the thymus. Thymosin and thymopoietin are hormones produced by epithelial cells of the thymus, which along with cytokines like IL-7 mature thymocytes into T cells. It is in the thymus that TCR develops. T cells reactive to self-antigens are destroyed. Pre-T cells reach thymus from the bone marrow and proliferate in the subcapsular region. Thus, CD-8- cells proliferate into CD4-8+ cells and reach the cortex region. Cortex contains 85% of the total thymocytes. By the time these cells reach medulla region, they have functional TCR and are either CD4 positive or CD8 positive. T cell precursors spend up to 7 days in thymus in differentiation. Two-stage positive and negative selection rescue thymocytes from apoptosis. The positive selection originates through interaction with MHC molecules on the epithelial cells of the thymus. Only CD8 positive cells which interact with an affinity within a critical range survive. Those with too high or too low an affinity to MHC molecules undergo apoptosis. During the negative selections, those binding to self-antigens undergo apoptosis. At this stage, one of the surface marker is downregulated and T cells are either CD4 positive or CD8 positive. The negative selection involves mostly dendritic cells at the cortex medular junctions and macrophages, which are present in both cortex and medulla. To summarize this lecture, we learnt about certain intracellular pathogens require cellular immunity for fighting against them. Even these pathogens have to deal with innate immunity. Many cells, of which TC and NK cells are prominent examples, they provide cytotoxicity for the infected cells. Cytokines play a key role in these cytotoxic processes, and thymus is the site where T cells proliferate and mature. With this background, we are now equipped to understand why TCR is so complex and how it develops during thymic education. However, before that, we will learn about the various types of T cells and MHC proteins. As we have briefly seen, MHC proteins play an important role in T cell maturation. Thank you.